Hello, let's start with the life cycle of Taenia solium. So, Taenia solium is a tapeworm. And why is it called a tapeworm? Well, because it resembles a tape. Exactly like a tape, it's long and ribbon like, tape like, so that's why it's called a tapeworm. So, let's say there is a Taenia solium inside the small intestine of a human being. Well, specifically in the jejunum of a human being. Taenia solium inserts its cirrus into the vagina of the same or different proglotid. Why? Because the presence of more than one parasite inside the body of a same human being, well, that is a different case, that is a difficult case, and the chances of that happening are quite slim, so that is not going to happen. Hence we are going to say that cirrus is going to be inserted into the vagina. Now what is cirrus? Here you can see cirrus is this part. This is the male copulatory organ, so this get inserted into the vagina either of the same proglotid or of different proglotid. Then zygote will get formed when egg and the male gamete combine. Zygote will then move into the ootype, which leads to the association of zygote with a yolk or vitelline cell, which exudates a chorionic membrane. So what happens is, well, first of all, the male gametes are here in the seminal receptacle, then female gametes are here also, then they get fertilized, then they move to, well, it moves to the ootype. Ootype is sort of a place where a lot many things come together, right? So eggs come and everything comes. So the ootype, in the ootype you have well, an attachment through vitelline duct to the vitelline gland. Now that is going to secrete a yolk cell, which then will form a chorionic membrane. So now we are over here. So zygote has formed, a chorionic membrane has formed, then the egg is going to pass into the uterus, which is lubricated by the secretions of Mehal's gland. So now what happens is, the egg is going to move up here, and these are the tiny egg capsules, are the zygote that are housed inside. Well, what is this? This is a uterus. Then inside the uterus, a holoblastic and unequal cleavage happens. What is holoblastic? As the name suggests, holoblastic is when one thing divides into two equal halves. But, well, two halves, but these are not equal. Hence the unequal cleavage. So unequal cleavage of zygote results in the formation of a megamere and an embryonic cell. Megamere will then form more megameres, whereas embryonic cell, well, it will form one micromere and one mesomere. So now we have three types of cells: megamere, mesomere, and micromere. These will get clumped together in sort of a micromere, mesomere, and then megamere fashion, and that is going to form a morula. So morula has inner micromere, which are sort of a round clumpy mass, median mesomere, and outer megamere. The Yuki cell, remember we were talking about the Yuki cell over here. Well, that is going to transfer its yolk to the megamere, and now the megamere will form a syncytial yolky fatty layer, which are going to nourish the forming morula. So the mesomeres, which are underneath the megamere, these will form a thick cuticularized radially striated shell called as embryophore. So now we are here. So the yolk that was present is going to transfer its yolk into the megamere, and then megamere, well, it is enclosing a mesomere. When the megamere disappears, the mesomere is going to form a cuticularized radially striated shell. Now you can see over here, it is a cuticle, it is a thick shell, right? Thick layer. This is the embryophore. Now what happens is, Morula, it develops three pairs of chitinous curved hooks at its morphologically posterior end, which are secreted by oncoblast. And this thing now, it's called as hexacanth. Hexacanth, along with its membranes, are basically oncospheres, right? So you can see curved chitinous hooks over here, a thick embryophore over here. So this becomes the hexacanth embryo, and the hexacanth embryo, along with its membranes, well, that is oncosphere. So now what happens is, by the time oncospheres are there, the proglottids inside the body of a tapeworm, well, they have turned gravid. Well, in order to understand what proglottids are, let's look over here. So when this is a Taenia solium, right? So this is a scolex, which is the head region. There will be a short neck, and then there will be proglottids. All of these are proglottids. These proglottids are immature. Why? Well, they are very young, they are immature, they are young proglottids. Then there are going to be these proglottids, which are mature proglottids. These mature proglottids, these are responsible for this fertilization event, because here both male and female reproductive system are present, and they are going to, produce zygotes and all of that. Now, we talked about zygote undergoing holoblastic and unequal cleavage. Now, by the time all of this has led to the formation of oncosphere, gravid proglottids have formed. What are gravid proglottids? There is no male, no female reproductive system, only the uterus has left. Right, uterus is left. So this is the uterus now. Can you really look at it? This is a uterus. So what is a uterus? Uterus is this. It was housing so many zygotes, and it was spread across the whole of the area, actually not whole of the area, but towards the interior of a proglottid. And now everything has disappeared. All of this, everything, the testes, everything has disappeared. Now what has left is uterus. So uterus is housing so many zygotes, the oncospheres, and apolysis happens. So what is apolysis? Apolysis is the detachment of these segments from the body of a tapeworm. Now, in case of Taenia solium, this happens in the bunches, like in the 4 or 5 bunch. Whereas in case of Taenia saginata, this happens singly. A single proglottid gets passed down with the stool. Now, when you look at the symptoms of teniasis, patients say that they feel somewhat irritated, or somewhat like when they pass stool, they feel some sort of sensations, because that is happening because the whole of the proglottid is getting passed down with stools. And when these proglottids leave the body, eventually they will get degenerated, because they are very fragile. And whereas the oncospheres, well, they remain, because remember, these have thick embryophore cuticularized layer. So let's say there is a pig roaming around, pig is eating food, grass and everything, and they live in close association with humans. So humans and pigs are living in close association with each other, and that pig encounters the feces of a human being, and it starts grazing upon it. Maybe it's mixed with food, maybe it's mixed with grass. And what happens is, the oncospheres, they enter the body of a pig through mouth. So this is the oral fecal route through which it enters. And then we have the oncosphere. It loses its embryophore and the basement membrane by the action of pepsin and HCl, which is highly corrosive. So when the oncosphere loses its two of the membrane, the hexacanth is liberated. The hexacanth moves down the small intestine, where it bores the epithelium of the small intestine with the help of its hooks and penetration gland. Now, hooks are for the mere attachment to the epithelium, whereas penetration gland, well, it does most of the work. Through this path, it reaches liver, through the portal system, arterial circulation, and finally the striped muscles. Striped muscles of maybe tongue, of maybe leg, basically striped muscles, because that is usually the part that humans feed upon, right? Because then let's say a human is eating the striped muscle of a pig, which is called as measly pork because it has all the uh, well, let's discuss what it has. Well, when it reaches the striped muscles, the parasite, it develops into a cysticercus larva or bladder worm. It's also called as cysticercus cellulose or the hyatid larvae. This is, well, this is the cysticercus larva. What it is like a globular structure, you can see, right? And it has this part. What is this? As you can see, this this part is the proscolex, because remember we were talking about scolex, and we said that scolex is the interior most part of the tapeworm, and it has hooks also, hooks and suckers also for the attachment to host body, etc, etc. So over here you can see that inside the pig the scolex is not developed, it's a proscolex. It's inverted, and it has suckers, it has hooks. Basically what an adult tapeworm has, it has all of that, but the catch is the proscolex is inverted. And it is called as bladder worm because there is a cavity sort filled with watery fluid, and that constitutes a bladder, and it has a thick bladder wall. So you can see over here the dimensions are written. It is 10 millimeter long, 5 millimeter wide, and the scolex remains invaginated. Let's say a human feeds upon the measly pork now. The measly pork enters the body of the human, the proscolex everts out, and the development of an adult worm takes place in about 10 to 12 weeks. And after this, the Taenia solium will start producing, well, it will start reproducing, it will do all the copulatory events, then whole of the fertilization and the development and everything. So that is in and about the life cycle of Taenia solium. So we discussed Taenia solium, we looked at the diagram of a cysticercus larva, a hexacanth larva, we looked at the immature proglotid, and this is a gravid proglotid. You can see over here that immature proglotids, well, these are very congested in structure, these are very small in structures, you can see so many of these over here, whereas gravid proglotids, these, as you can see, have these uterus, the lateral walls of uterus, and in case of Taenia solium you will see 9 to 13 lateral branches of uterus, whereas in case of Saginata there are many more. And this is the adult tapeworm. You can see over here scolex, or the anterior most part of the tapeworm, it's quite small. This is almost a knob like structure, right? So that is the anterior most part, and it has hooks, it has suckers. All of these help in the hosts, in the parasite's attachment to host. And yeah, that's about it. Thank you so much for watching. I hope I see you soon.